and welcome. My name is Naqing. I'm from Clemson University Center for Human Genetics. I'm happy to share with you my current project, pipeline building for integrating unpaired single-cell RNA-seq data and single-cell ATAC-seq data. So we all know RNA sequencing can measure the gene expression. There are transcription factors can regulate gene expression by binding on the DNA region like enhancer and promoter. So the DNA region, if it's open for the transcription factor to bind on or not, it's called transcription chromatin accessibility. This could be measured by the ATAC sequencing, but in the traditional way, this two-difold technique can only be done on different cells, which generate unpaired single-cell data. So to infer the gene regulation, how to integrate this unpaired information of gene expression and chromatin accessibility. This is called the diagonal integrating problem. When we want to integrate this, we can see the data. This looks like this: ATAC-seq and RNA. You can see neither the cells nor the features are matching. So the first thing we do is find a way to link the features. Many methods calculate the gene activity score, which is basically predicting the gene expression from the ATAC-seq data, and then now the two data all have gene features and easier to integrate. And there are other methods use different approach like guidance graphs. The next step is dimension reduction. Basically, because the data is sparse and high dimensional, we reduce the dimension to lower dimension. And then this is very crucial steps, and many approach has been developed to do this. And then next thing is calculate the cell similarity based on the embedding and the clusterless cells. In this step, we can also do the transfer label, which means we first cluster the RNA and then transfer the label from RNA to the ATAC. And also we can do jointly labeling, which means we jointly give the label for RNA and ATAC-seq data. So as I say, for many, each step have many choices, and it's challenging to choose the optimal one. And at the beginning, there was no ground truth, and those many methods have not been evaluated with the ground truth data systematically. And recently, the paired data is available. It's a great opportunity to do the evaluation now. So what I did is I evaluate the methods at each step, including gene feature linking and basically gene activity score, and then dimension reduction and also labeling. Not only compare between the different methods at each step, but also look at the different combination between each adjacent step. Some of the combination have already been developed as famous integrating methods, but the other combination haven't been tested yet. There are some results. First one is gene activity score. Here it shows the Pearson correlation of gene activity score calculated by different methods with the real gene expression. Here x-axis is different method and the y-axis is Pearson correlation. Here I only show the PBMC data's result. But the other two data have similar results. So the Pearson correlation is generally low, although on highly variables, slightly higher. For embedding evaluation, I looked at for each RNA cell, if I list the 100 nearest neighbor from ATAC cell embedding, if I can find the corresponding cells in this 100 nearest neighbor. This paired data. So I looked at the percentage, and we can see glue have high performance on PBMC data but for other data it's not stable. But the UniPort is most stable and good performance on this metrics. For the next evaluation metrics is cell type average silhouette index. Silhouette index is a measurement to evaluate the embedding with the label. So if the silhouette index is high, it means the cell in the embedding are similar to each other when they have same label, and the cells have different label. They are also distinct on the embedding. So you can see here, x-axis different method, y-axis is the mean silhouette index of the embedding, and here the triangle is ATAC embedding, and round dot is for RNA embedding. Different color means different data set. Here, acid joint have higher silhouette index on the RNA embedding, but for acid joint have RNA label requiring, so I think this is not a surprise. For lastly is labeling evaluation. I used NMI and ARI, which is the commonly used measurement to measure the consistency of two label. So I split to two group, which is transport label and jointly clustering group. For transport label, I choose CIRAD and acid joint. We can see the CIRAD is outperforming, and for joint clustering, I choose the different embedding and did the simple low-in clustering. And we can see glue and the Uniport have better result, but overall the transport label have better result. And lastly, I want to thank my co-authors, Dr. Yan and Dr. Dana Durin, and the other Dr. Durin lab members, and the bioinformatics core in the CHD.